Hi everyone, and welcome to today's screencast. Today we're going to be talking all about nucleic acid. The nucleic acid is a molecule that most of the time you find in the nucleus of a cell. And of course, I'm talking about DNA and RNA. Both of those molecules are very large. DNA in particular is the largest molecule in our bodies. Now, you'll need to know that DNA stands for deoxyribonucleic acid. And RNA is ribonucleic acid. Now DNA, you can see in this picture right here, you'll see it has two sugar phosphate backbones, and those backbones are held together by the bonds that form between the letters that we always refer to when we talk about DNA. T stands for thymine, A, which stands for adenine, C, which stands for cytosine, and G, which stands for guanine. When we're talking about RNA, on the other hand, RNA only has one sugar phosphate backbone. Here. And you'll notice that RNA looks a lot like the alpha helix of a protein. The reason we know it's not the alpha helix of a protein
is because, again, you're going to see those letters A, C, T, and G, so you'll know it's an RNA.
So nucleic acids are composed of nucleotides, and you'll need to understand how a nucleotide is put together. There are three parts to a nucleotide. The first part is a pentose sugar. Pentose just means that it's five-sided, so it's a sugar, looks almost like a house. And if it's in RNA, then it's a ribose sugar. If it's in DNA, it's a deoxyribose sugar. So here's the ribose sugar and the deoxyribose sugar. You'll see the only difference between them is that ribose has an extra oxygen. And that's why we call DNA deoxy. It's got one less oxygen, ribonucleic acid. Second part that you'll see in every nucleic acid and every nucleotide is a phosphate group. Now we've already talked about phosphate groups. CO4, CO3. The phosphate group has a double rotating bond, and that's the part of the nucleic acid that allows the chain to twist and bend into a twisted double helix. The third part of a nucleotide is the nitrogen base, and a base can be either purine or a pyrimidine. So a pentose sugar, a phosphate group, and a nitrogen base. So what's the difference between a purine and a pyrimidine? Let's find out. If you look at this nucleotide, you'll notice that the nitrogen base is a pyrimidine. What's the difference between a pyrimidine base and a purine base? Well, you'll notice that a pyrimidine only has one ring on its structure. So you have a phosphate group here, you've got a sugar here, and then you've got this base that only has one ring on it. If you look at a purine, a nucleotide with a purine, you'll notice again, here's the phosphate, the sugar, and here's the nitrogen base. It's a purine because it has two rings attached to it. So which letters, A, C, G, and U, are purines and which are pyrimidines? Let's talk about purines first. So purines, again, have 1, 2 rings in their nitrogen base. And the only two letters that do have two rings are adenine, represented by the letter A, and guanine, represented by the letter G. You'll see that adenine has two rings and guanine has two rings. So adenine and guanine are both purines. They have two rings. And the memory trick I use to remember is it's got to be gap. Why 2? Because two rings. Gap, guanine, adenine are both purines. It's got to be gap. Anything with 2 rings, gap. Now, you find adenine and guanine in DNA, and you also find adenine and guanine in RNA. Pyrimidines, we already know, have only 1 ring structure. And there's 3 different nitrogen bases that are pyrimidines. First of all, thymine. Thymine's got 1 ring. It's represented by a T. You only find thymine. Uracil. Uracil also has 1 ring, represented by the letter U. And you find uracil in RNA only. Cytosine, represented by the letter C, 1 ring only, and cytosine you find in both DNA and an RNA. Cytosine, thymine, and uracil are all pyrimidines. They have 1 ring. And the memory trick I use for this to remember that these are all pyrimidines is cut, C-U-T, cytosine, uracil, and thymine, cut the pyramid. Pyramidines. If you cut the pyramid, you'll only be left with 1 ring because you cut. So there's less than. So there's only 1 ring in cytosine, uracil, and thymine. Cut the pyramid. That's my memory trick. If you're going to break that down and you're comparing DNA and RNA, the pyrimidine only found in DNA, and the uracil is only found in RNA. All the other letters, A, G, and C, are found in both DNA and RNA. The sugar that DNA has is one less oxygen, so we call it deoxyribose, found in DNA only. And the sugar in RNA is different with more oxygen. It's called ribose, found in RNA only. And of course, both DNA and RNA have a phosphate group. Now, how is DNA put together? Well, when you join the letters together, you have to use dehydration synthesis, and you lose a water every time you join 2 molecules together to form a bond. And DNA is composed of 2 strands that are joined together. And those 2 strands are joined together via hydrogen bonds. So adenine always joins with thymine. And the way that they link together form 2 hydrogen bonds. And cytosine always binds with guanine and vice versa. And they bond with 3 hydrogen bonds. So adenine binds with thymine with 2 hydrogen bonds. Cytosine joins with guanine with 3 hydrogen bonds.
Okay, so if you look at guanine and cytosine, you'll notice that it has 1, 2, 3 hydrogen bonds holding it together. And so there's a memory trick for that. Think crazy glue. C for cytosine, G for guanine. Crazy glue because it takes more glue to hold them together. It takes 3 hydrogen bonds. Now, if you look at adenine and thymine, you'll see it takes 1, 2 hydrogen bonds only to hold together. And my memory trick for that is at. At is a 2-letter word, and so you only need 2 hydrogen bonds to hold A and T together. Now here we can look at everything all put together. Here's guanine and cytosine held together with 3 hydrogen bonds, and then you look at thymine bound to adenine with 2 hydrogen bonds. So A is always with T, C is always with G, and when you look at them together, those sugar phosphate backbones will then twist into a double helix. So deoxyribonucleic acid, DNA, is made of the 2 linear strands of the nucleotides joined together, and like I said earlier, they join as a twisted ladder, as a double helix. So what does DNA do? It controls everything that happens inside the cell, and the way that it does that is by telling the cell which proteins it needs to make. The second thing that we know about DNA is it has all of the genetic information that it needs to make a whole new you, a whole new organism. So every single cell in your body has exactly the same DNA. And what that means is that a heart cell if we could ever figure out how to do it, has the DNA required to make a brain cell, and vice versa. Now let's talk about RNA for a little bit. RNA is made by DNA, so RNA is really just a copy of one section of a DNA molecule. What's different about RNA is that it doesn't have to stay in the nucleus. It can actually move out of the nucleus and go into the cytoplasm. So you see here the nuclear pore, the nuclear envelope, and the RNA can go outside through a pore, and then it's going to go out and it's going to find itself a ribosome. And the ribosome will help the RNA make a protein. Now we already have talked about the fact that RNA has a ribose sugar instead of a deoxyribose sugar. That's that pentose sugar we were talking about. And RNA also has uracils instead of thymines. RNA is single-stranded. It doesn't make a double helix, so it looks a lot like an alpha helix of a protein. And there are three types of RNA. There's mRNA, tRNA, rRNA. rRNA stands for ribosomal RNA, tRNA stands for transfer RNA, and mRNA stands for messenger RNA. And all of those basically help DNA to make proteins. So DNA, right here, makes a copy of itself as RNA, and then the RNA goes out and finds a ribosome, and ultimately what's made? A protein. So when you compare the DNA and the RNA, DNA and RNA both have phosphates. They both have a sugar, deoxyribose versus ribose, and they both have a base. That's the nucleotide. Now the bases are different, only that DNA has T, RNA has U. They all have A, C, and G. And then lastly, DNA is a double helix, whereas RNA is just a single strand. Now the last thing we need to talk about is ATP. Now, ATP is adenosine triphosphate, and it is also a nucleic acid. And the reason we know it's a nucleic acid becomes very clear when you look at the parts of ATP. ATP has 3 phosphate groups, and it's attached to an adenine, just like in DNA and RNA, a ribose sugar, just like in RNA, and then instead of just one phosphate group, it has 1, 2, 3 phosphate groups. And you'll notice those bonds are extremely high energy bonds that hold those phosphates together. And that's where we get energy for our body. Now we already know that the mitochondria makes ATP, and it does that by turning sugar, glucose, into ATP. And the reason that there's so much energy stored in ATP is because it takes a lot of energy to put 2 of those negative phosphate molecules together. So when you use a lot of energy to force those together, that's a high energy bond. And then if you do that with a third phosphate group, it's an extremely high energy bond. And if you break that bond, lots of energy gets released. And so make sure that you can memorize cellular respiration. Sugar, glucose, joins with oxygen to make CO2 and water, 2 waste products, and ultimately energy in the form of ATP. Heat is also released in the process. So make sure you come to class with all of your hot questions all about nucleic acids.